There are two items. One is measurement of ESR. How ESR is determined and its usefulness. And another is reticulocyte determination, or what is its number in circulation, how it is, how we count reticulocytes. That will be taught. This is for only these two, ESR and reticulocyte count. It is there in the syllabus, but you are not to perform these experiments. Only thing for viva voce or in exams purpose you may require it. For example, what are the conditions in which ESR is raised? What are the conditions, disease condition or physiological conditions in which ESR is lower? ESR means erythrocytic sedimentation rate. Erythrocytic sedimentation rate means if you take some amount of blood which is having anticoagulants in it, that means the blood will not be coagulated. And you are taking in a tube some blood having anticoagulants in it so that it doesn't get coagulated. So if I put and keep this blood for 1 hour, okay, we will find the RBCs are settling down. RBCs are settling down. And at the top, there will be plasma. So this rate of settling down of RBC is called erythrocytic sedimentation. Erythrocytes are sedimented. So this process is called erythrocytic sedimentation. And the rate in 1 hour, how much this is separated. So this is called erythrocytic sedimentation rate. Actually, we are not measuring the how much RBCs has been settling down, but how much clear fluid we are getting. That is actually measured. With this measurement, suppose this plasma, separated plasma is much more, that means sedimentation rate is high.
Yes.
So in that way, we measure ESR. In different disease conditions or in physiological conditions, this may be raised, this may be lowered. So for that purpose, it is important. You have to know that. Okay? And another is reticulocyte. So reticulocytes also can increase. No question of decreasing. It remains normally 1% out of 100 RBCs. Only 1 RBC will be available in the circulation. But when the demand of RBC is raised, demand of hemoglobin is raised, then what will happen? When there is increased, when demand becomes increased, what is the condition? When we demand more hemoglobin, when we demand more RBCs, when? When we feel anemic, when we feel anoxic, hypoxic. So it will be stimulated, there will be liberation of erythropoietin that will cause increased erythropoiesis. So increased rate of erythropoiesis, since the demand is high, so immature RBCs, that is reticulocytes, will be coming out in circulation. So it may be 3%, 4% at the time. So if we find 3%, 4% like that reticulocyte in blood, that means what? Increased rate of erythropoiesis is going on. So that is why it is done. Okay. And another thing, when there is increased rate of reticulocyte formation, means coming in, it indicates my bone marrow are now active, are now very active. They are actively producing erythrocytes. So that's why this is important to you. Okay, so this class on this will be tomorrow. It will be taken by I will be there. It will be taken by other, those who are there. 3 teachers are there. They will be, I will be remain present. They will be telling you what is the significance, how it is measured, what are the procedures. So you try to learn that. So during examination, you may be asked, though there is no practicals on these, but you may be asked how it is measured, how reticulocyte is measured, how erythrocytes are measured. Okay? So today I will start general physiology. From today I'll start general physiology. You go through the topics of general physiology. So first topic is on cell. Cell is structure and function. All of you are having much knowledge regarding this. Even then, I'll go through it. So cells are what? Okay. So what are there within the cell, in a cell? So many things. So I can classify them. One is nucleus, another part is cytoplasm, and another part is cell membrane. Okay? So what is there in a cell? Cell membrane. Within the cell membrane, you will get cytoplasm and nucleus. Let us see what the cell membrane looks like and what about its structure and what about its function. Okay, we know the structure of cell membrane is described by fluid mosaic model. Okay, fluid mosaic model. We'll see what is this fluid mosaic model. And chemically, what are the constituents in a cytoplasm? Chemically, what we will find? Lipid, protein, and carbohydrate. What are the constituents? Fine. Lipid, protein, and carbohydrate. Lipids are of 2 types here. 3 types rather. Phospholipid, glycolipid, and cholesterol. What are phospholipids? What are glycolipids? What are proteolipids or lipoprotein? What are lipoprotein? What are lipoproteins? When lipid remains combined with protein, we call it lipo. So it is the compound of lipid and protein. So they are lipoprotein. And glycoprotein, glyco means carbohydrate, protein and carbohydrate, glycoprotein. Phospholipid, lipid and phosphoric acid. Here we will find mainly phospholipids. Here we will find phospholipids. Moreover, there we will find glycolipids, means lipid with carbohydrate, lipids with carbohydrates. So these are and plus we will get cholesterol. Cholesterol is important for cell membrane. We know the fluid mosaic theory hypothesis, it tells this is fluid, it has got fluidity, it is not hard. And not only this, these are not fixed, they are moving. The lipid layer is moving, and within this lipid bilayer, there are some proteins. There are some proteins immersed in the bilayer. Okay, different types of protein. Have you seen mosaic floor?
Yes.
How it is?
It's plain and they are drop.
What?
Mosaic.
Mosaic. Okay. Cemented structure, and in between there are mosaics. Colored stones. Okay. Different colored. So here also you will find cemented structure. Cemented structure, I mean the lipid bilayer. So on this lipid bilayer there are mosaics. What are these? Proteins. And mosaic has different colors. So here also we will find different proteins. Some proteins are present from outside to inside. Transmembrane. These are transmembrane proteins. And some proteins you will get only on the outside. Some part of this protein is immersed within the lipid bilayer. Similarly, the other side also may contain this type of protein. So some are eccentric, some are intrinsic proteins. We will discuss. A cell is the smallest unit that is capable of performing functions. Okay? All living things are made up of cells. No doubt about it. Cells are the smallest working units, no doubt about it. And all the cells come from pre-existing cells. Obviously, through cell division we get it. These are some examples of cells. And cell parts. In a cell, the molecules, biomolecules are, what are the biomolecules? Nucleic acids, DNA, RNA. So these are nucleic acids, lipids, carbohydrates. And these structures, these compounds make membrane, organelles. Okay, so let us see. Organelles are actually, what are organelles? Structure that are membrane bound. See mitochondria, it is having membrane. Endoplasmic reticulum having membrane. Golgi body is having membrane. Lysosome having membrane. So there are membranous structures. Arrangement of cells. So cell is having protoplasm. So protoplasm, what is? Cell inclusion and cell. Protoplasm bound by the cell membrane. Everything including the nucleus, these are protoplasm. Okay. So in the protoplasm you will get cell inclusions and cell organelles in the cytoplasm. You will get cell organs and cell inclusions. So what are these cell inclusions? Can you give examples? Very simple.
What's the question, sir?
Can you name some cell inclusions? What? Tell?
The vacuum.
No. Cell inclusions means they are not membranous. They are particles, molecules. A cell needs glucose. Where from it is getting glucose? From outside. It is coming by means into the cytoplasm. So these are, we know the cell may have glycogen. How these glycogen are formed? By joining glucose molecules. So it may contain glycogen. Cell want to produce some proteins. Proteins are, cell will produce these proteins. So raw material is amino acids. Where from it will get these amino acids? From circulation. So from blood it has to enter within the cell. So first it will enter into the cytoplasm. So we will get this. Similarly, we will get fatty acid, glycerols, vitamins, oxygen, carbon dioxide. So these are present some enzymes, free enzymes. We know glycolysis takes place in cytoplasm. So to conduct this glycolysis pathway, to conduct the reactions of this glycolytic pathway, so there some enzymes are required. So where from these enzymes come? They are also present in cytoplasm. So in the cytoplasm, you will get enzymes also. You will get ribosome, RNAs also. Nucleic acid RNA you will also get, because protein synthesis, when mRNA will be passing to the endoplasmic reticulum through the rough endoplasmic reticulum. Rough means there will be ribosomes. When mRNA will be passing through these ribosomes, ribosomes, they will read it. I have to make a peptide chain. The first one is glycine. So this is present in the cytoplasm. But who recognizes glycine? So there are some RNAs who knows glycine. So we'll go and collect the glycine and bring it to the endoplasmic reticulum. Okay, this is your glycine. Next, for the synthesis of the polypeptide, next is say arginine. So arginine is known by other RNA. So he will find out within the cytoplasm where is arginine. So it will bind with that and bring it to the endoplasmic reticulum and will supply to the ribosomes. Ribosome will bind the glycine, previous one, with the arginine and through a peptide bond. So next, in this way, a lot of amino acids are coming and joining. So that's why this should be present within the cytoplasm. RNAs, these RNAs, tRNA, transfer RNA, they are called transfer RNA. So they should be present. There are sRNA, soluble RNA. So they are also remain present in the cytoplasm. So in the cytoplasm we get so many chemicals, so many compounds. We may get excretory products also. When amino acid proteins are catabolized in the body, so ammonia is formed, immediately this ammonia is converted into urea. So this urea will be first coming to the cytoplasm, so you may get this. So excretory products you can also get in the cytoplasm. Clear? So beside it, there are a lot of electrolytes in the cytoplasm. Sodium ion, potassium ion, chloride ion, bicarbonate. What is their functions? What are their functions? These ions?
Equilibrium.
I touched you. How could you feel? I touched you.
Nerve impulses.
Nerve
signals.
He said, I touched and something happened in this area and it has created a sensory impulse. So this is carried through the nerve to my cerebral cortex and there are sensory areas. So they identified, somebody has touched. Who has touched? This I have seen him. So sir has touched me. Okay, so in this way we sense. If I pinch, there is pain. It is not the touch, it is pain. So there are some different receptors for pain. If I place some hot water,
Pain receptors.
not the same receptors. There are some thermoreceptors. If I place some ice, there are cold receptors. This is neither pinching, neither touching. What is this? Pressure. So there are some mechanical receptors on the skin. How these receptors understand means the receptors are disturbed and they are producing action potential. And how these action potential are made? Action potential is having the characteristics of when action potential is produced, it is having it can travel. It can travel along. So this receptor attached to the nerve fiber. So this action potential will be as sensory impulse. This will travel along the nerve fiber. Now, hello. Let us say, I want to explain the role of these ions. Sodium ion, potassium ion. You have the syllabus resting membrane potential, action potential.
Sir, the hot water you said, that is also pain receptor.
Which one?
Sir, hot water.
No. There are separate receptors.
And the cell inclusion you said, there is a nucleosome and peroxisome chemical.
Yes, they are also there. These are also there. So many things are there. So many things are there in cytoplasm. Similarly, there are internal pH has to be maintained. So some buffer should be bicarbonate ion, hydrogen ion, ammonium ion. So they should be present within to maintain the pH of the proteins. The cell proteins are synthesized in the endoplasmic reticulum. They will be, after synthesis, they will be coming to the endoplasmic reticulum. So there it will be formed, or some vesicles will be formed containing these proteins, and they will be liberated from the endoplasmic reticulum into the cytoplasm. And from the cytoplasm, it is required by other cells, not these cells. So it will be exocytosed. Some proteins are not membrane bound, not in the vesicular form. They are directly released from the endoplasmic reticulum into or from the Golgi bodies directly into without any covering, packaging, they directly come into the cytoplasm. Like enzymes. The enzymes are also produced in endoplasmic reticulum. That's why you will get these enzymes. Cell, outside the cell membrane is normally polarized. Polarized means both the charges are there. So cell membrane normally, particularly the excitable cells. Can you name the excitable cells? Excitable cells. They show the property of excitability. Some cells show the property of contractility. Muscles, they show contractility. Before it shows contractility, it must have excitability. After excitability, then only it will be contracted. So, muscles are excitable tissues. Nerve cells, axons, they are also excitable. Neurons, they are also excitable tissues. So, suppose this is an excitable tissues. So, they are polarized. We know how they are polarized inside and outside of the cell membrane. Outside is positively charged, inside is negatively charged. So now we say the cell is polarized. How this polarity developed? This polarity is developed by sodium ion, potassium ion, chloride ion, these ions. Under normal condition, you will find sodium concentration, higher concentration of sodium will get outside the membrane. Inside the membrane, you will get less sodium ion. So there is a concentration gradient. So we know there may be diffusion. Since concentration is higher outside, so this from higher side, sodium ion should enter within the cell. The problem is, cell membrane will not allow this. You are not permitted. You are not permitted to enter. But it allows when the cell membrane is disturbed by a stimulus. It may be mechanical one, it may be thermal sensation, it may be electrical sensation, it may be a chemical sensation. Chemically, I can excite a cell. So there are many types of stimulation. So whatever the kind is, this stimulation is capable of changing the permeability of the cell membrane. As a result, permeability changed. There are ions channels. You will find there are certain proteins. They are actually performing transmembrane proteins. देखो So these transmembrane proteins are channels, ion channels. So there are sodium channels. So when this is disturbed, sodium channels will be opened up and now sodium ion will enter. What will be the result? The internal negativity, it is entering within the cell with positive charge, but inside is negatively charged. So gradually, this polarity, under normal condition, since there is a difference in polarity, so it must be having a potential difference. Potential difference. This potential difference is called resting membrane potential, which is about minus 70 millivolt, depending on cell types. Think about well. Water level is here. So I am to bring water. So water level is at very deep. It means resting membrane potential is 90. Its excitability is less. When the water level is here, then it will be easier. It means its excitability, its resting membrane potential is minus 50. What happens when sodium ion enters? So it will minimize the negativity. So initially it was minus 70. So it will minus 65, minus 60, minus 55. When it comes to a particular level, say minus 55 or minus 50, that initially there was one door open, one channel was opened by the stimulus. So through this channel, sodium ion is entering and it is changing the resting membrane potential towards positivity. Minus 70, minus 60, minus 50, minus 30, minus 20, minus 10, then 0, then plus 1. Reversal of plus 10, plus 2, plus 3 or plus 40. So this is the reversal of potential. This is called depolarization. Polarity is changing. Polarity is changed. When this depolarization means when it comes to a particular level, suddenly lot of doors become opened. Lot of doors becomes open. Door means sodium channels. So now lot of sodium ion will be entering into the cell. So there will be rapid influx of sodium ion and it will cause development of action potential. And with this sudden movement of sodium ion, you will get action potential. At that time, potential is plus 30 to plus 35 millivolt. Plus, not minus. And this is called the action potential. Now this action potential, if it is, this cell is connected to a nerve fiber, now this action potential will be travel along the nerve fiber. And you will feel, I have touched. So for this purpose, sodium ion is essential. And okay, now it has produced this. So lot of sodium ion entered. Now what will happen to this cells? Action potential is done. Then it has to be brought back to the normal position. How? It has entered, sodium has entered here and caused depolarization and ultimately action potential. Now what will happen to this sodium ion? Still sodium ion is much more outside. So this sodium ion which have entered, it has to be returned to the outside. But how? By active transport. By sodium-potassium pump. Okay, so it will, the pump operates, exchanges sodium ion and potassium ion. It will send back 3 molecules and from the outside it will bring potassium ions. Ultimately net 1 positive charge is more. So during pump, again the concentration of sodium ion will remain same. Similarly, in development of resting membrane potential, there is role of potassium ion, there is inside is negative, there are role of proteins. Proteins can be depending on the pH we have seen, depending on the pH of the solution, protein can be positively charged, can be made negatively charged. So inside the cell, the pH is such that the proteins become negatively charged. The negative charge of proteins, negative charge of chloride ion, negative charge of bicarbonate ion, they produce the negativity. And the amount of positively charge is relatively less within the cytoplasm. So all these are responsible for creation of resting membrane. I will be explaining it later when this will be discussed, particularly in this unit, in this general physiology unit. So in this way, we can see what are the importance of these ions. Without these ions, the cells cannot be excited. It is the role of these ions. Moreover, you see, all proteins, all hormones are having their target cells. So suppose this is the target cell. This is the target cell. So it has to make certain changes within the cell. From blood, for example, this is TSH. And this is the cell of the thyroid gland. So the function of TSH is to stimulate the thyroid cell to produce more T3, T4 hormones, thyroxine hormones. So how it will produce? How it will produce? Or this is a muscle cell. And there are actin myosin filaments. When these will be stimulated, the actin myosin filaments will make cross bridge and contract. So for this purpose, it has to be stimulated. Now they take the cases of these to produce some effect on the cell. the cell membrane should have receptor proteins specific for TSS. Specifically for TSS. Only TSS can bind with these receptors. So this will bind with these receptors, and these receptors on the other end this is the receptor, here is the binding site, and here is the catalytic site. When it binds with this, immediately there is some changes on this site, catalytic site. Because this is made up of these enzymes get stimulated. When this is produced, means second messenger, they will produce the second messenger. Action of this will produce second messenger. Say cyclic AMP example, and this cyclic AMP will produce lot of change. Because of this change, there will be increased production of thyroxine, T3, T4. Thyroid hormone biosynthesis will be stimulated.
So what you will do? So what you will do? Exactly.
This one, second messenger. So this is also a transmembrane protein. Receptors also transmembrane, like channels. Channels are also transmembrane proteins. These are the membranous structures. What are the membranous structures? Nucleus, mitochondria, endoplasmic reticulum, Golgi body, lysosome. And non-ribosomes, chromosomes, centrioles. This is a typical animal cell. So what you will be having within the cell, other than cytoplasm, some organelles. What are these organelles? Mitochondria, Golgi bodies, lysosomes. So these are the organelles. Surrounding the cell, what is there? Cell membrane. So this is a part of the cell membrane. This is the lipid layer, bilayer. This is one layer, this is another layer. In between you can see different proteins. So there is double layer of what? Lipid. That's why we call lipid bilayer. Let us understand what the structure of this bilayer. See, this is the lipid bilayer. This is one phospholipid. This is phospholipid. These are phospholipids. Okay? These are phospholipids. This is one layer and this is one layer. And in between you can see these are the cholesterol. Cholesterol renders flexibility. If there is more cholesterol, it is more flexible. Membrane is more flexible. Less cholesterol, it's less flexible, means it is rigid. Another thing, important compound that renders flexibility, it is the one layer, another layer. See, what is there? This is phospholipid, no? Phospholipid made up of triglyceride, not triglyceride, diglyceride. Glycerol. This is glycerol. All means alcohol. This alcoholic group can bind with acidic group through a bond. What is this bond called? Ester bond. These bonds are called ester bond. The bond between alcoholic group and acidic group is called ester bonds. So now, fatty acids can join. They are having fatty acids are all the fatty acids must have an acidic group, and these acidic groups are actually carboxylic group. So through the ester bond, through the formation of an ester bond, this can combine with a fatty acid. Now this is monosaccharide. Similarly, this can also bind with another fatty acid. Now it is disaccharide. When it combines with another fatty acid molecule, it is trisaccharide. So trisaccharide means there is glycerol and 3 fatty acids, triglycerides. Let us see how this is formed. No fatty acid. Now it is diglyceride. And this group may combine with phosphoric acid. Now it is phospholipid. Now it is phospholipid. Now this phosphate group can combine with choline. Acetylamine. These are nitrogenous compound. Acetylamine, sphingosine. You will find all these types of glycolipids, phospholipids, not glycolipids, phospholipids in the cell membrane, in the bilayer. Now see, these fatty acids, we know fatty acids are of different kinds. Some are long chain fatty acids, means number of carbon atoms is much more. And some are short chain fatty acids. Their length is less because of there is less number of carbons. So it may be short chain carbo acid and long chain fatty acids. Fatty acids may also be classified according to their importance, essentiality in my body. So some are essential, some are non-essential fatty acids. Essential means they are not synthesized in our body. So we have to take like essential amino acid. There are 3 essential fatty acids. So we have to arachidonic or arachidonic acid is one of the example. Okay, so they are important since they are not synthesized in the body, but they are important constituent of cellular constituents. Okay, so forget about this essentiality. Now let us see how we can classify. There is another further classification depending on the presence or absence of double bond in between the carbon atom. If there is double bond, means unsaturation. So these fatty acids will be unsaturated fatty acids. When there is no double bond, how double bond is created? These are the fatty acid carbon, and all the carbon are having hydrogen. So now all the valencies are saturated. So these are saturated. No double bond. No double bond. Okay? Now I am doing one thing. I remove this hydrogen atom and also remove this. Two hydrogen atoms are not there. Somebody is taken away. NAD or FAD. There is dehydrogenation. So because of that, hydrogen atoms are removed from there. What will happen? Array, no hydrogen is there. So they will now there are 4 valencies. 1, 2, 3, 4. 1, 2, 3, 4. Now in this way, unsaturation is made. There is double bond in between the carbon. When such double bond we get within the fatty acid chain, then we call it unsaturated fatty acids. It has got importance. Why? It may be unsaturated. It may be unsaturated. When there is unsaturation, more unsaturation means the membrane will show fluidity, more fluidity. And if more saturation is there, then it will show less fluidity. Understood? So this is the importance. So what does it mean? These are actually fatty acids. These are actually fatty acids. Fatty acids, fatty acid, fatty acid, fatty acid, fatty acids. See, fatty acids are fetching each other. Of this layer, fatty acids of this layer is fetched by the fatty acids of other layer. So they are fetching each other. And this part means which part? Non-polar part. This part is called non-polar part and hydrophobic. So this part is non-polar, no polarity is there, also hydrophobic. Means water soluble thing cannot pass through because of this layer. And it made the cell membrane semi-permeable. Okay, but those who are soluble in lipid, they can easily pass. No problem. Only water soluble particles, substances, they cannot pass. And for that purpose, if passing is essential, they have to follow the other means through either through carrier, transporter, or through channels like glucose channels is there, amino acids channels are there, water channels are there, ammonia channels are there. So there are different kinds of channels are there. So through they have to use those channels, but those channels are not always open. They are to be made made open, then only they can pass through. Glucose cannot pass through, though there is channels. So these channels can be opened only by insulin. So if there is no insulin, the channels will not be opened. Glucose will not be able to enter. So there will be raised blood sugar level in the blood. The amount of sugar, glucose will be more. This happens in diabetic patients. And this part, where it is, what is there within this part? There is phosphate, and attached to this phosphate, choline, sphingosine, or another, what are the name? Ethanolamine. Okay, so they may remain present there. And this part is opposite to this, means they are polar. They are polar and not hydrophobic, they are hydrophilic. That means this part is water soluble and the middle part is water insoluble. Okay? So in between we got cholesterol. We said what is the role of cholesterol. And you see, thickness 8 nanometer. This is what? Channels. This is what? A receptor. So here hormone will bind. Binding site is there. So it will cause stimulation of this part. A stimulation of this will produce second messenger. And this second messenger will make necessary changes within the cell. Hormones, these hormones, particularly the protein, the hormones which are protein in nature, they cannot enter because they are not water soluble, or fat soluble. They are water soluble, so they are not allowed to pass in. So they are allowed to bind with the receptor only. Then only it can produce the changes. But there are some fat soluble hormones. Examples? Example of fat soluble? Yeah, steroids. Very good. Steroid. Example? Estrogen, progesterone, testosterone. These are produced in gonads. And are there any other source other than gonads?
Adrenal.
Yes, adrenal cortex. Adrenal cortex. In the adrenal cortex.